Good morning, all my dear students. Today we are going to learn a very useful separation technique, electrophoresis. In this lecture, we should discuss the electrophoresis introduction, then the definition of this technique, who is the father of the electrophoresis, what is the principle involved in the technique, the types of the electrophoresis. The one of the type we will discuss, that is moving boundary electrophoresis. What are its advantages, disadvantages, and the capillary electrophoresis. What will be the learning outcome of this lecture? The students will be able to understand about the separation technique, electrophoresis, with its principle, types, definition, as well as the moving boundary and the capillary electrophoresis method. What is electrophoresis? Electrophoresis is a physical method of the analysis which involves the separation of the compounds that are capable of acquiring the electric charge in conducting electrodes. So this is a technique useful to separate the molecules which is having the charge or acquires the electric charge in the conducting electric field. So for those samples, the electrophoresis method is used. Many of the biological molecules like amino acids, nucleic acids, peptides, all possesses the ionizable groups. Means they are having charge on it. So when we apply the external electric field to those molecules, then they start migrating towards the opposite charge electrode, and on the basis of this movement or the migration, the molecules can be easily separate out. So here come the definition. Electrophoresis may be defined as the migration of the charged particle through a solution under the influence of the external electric field. So when a solution just provided the external electric field, it causes the charged particle of the molecules in a solution to migrate towards the opposite end of the electrode. So ions that are suspended between the two electrodes tends to travel towards the electrode that bears the opposite charge. So on the basis of this migration and the movement, the sample can be easily separate out, and the molecules, the charged particles differs in the size, shape, as well as their charges are also having the difference. So on the basis of their difference, the migration speed also differs. And when the migration speed differs, some of the ions move faster and reach to the electrode first. Some are slower and reach to the electrode in a longer period of time. So on the basis of this method, it can easily separate out the charged molecules present in the solution. Who is the father of this wonderful separation technique? The father of the electrophoresis is Arne Tiselius, Sweden scientist. Won the Nobel Prize in the Chemistry 1948 for his research on electrophoresis and adsorption analysis, especially for his discoveries concerning the complex nature of the serum proteins. This type of the cell is essentially a bent glass tube with the electrolyte reservoir. This is the tube you are able to see, containing the cathode and the anode electrode and a buffer containing the macromolecules that need the electrophorist. He tested the horse serum in the apparatus and found four distinct bands consisting of albumin, three globulin, which he named alpha, beta, and gamma. So this is a bent glass tube in which the electrolyte reservoir is there with having the two electrodes with a buffer solution, and the alpha, beta, gamma proteins are to be separate out from the horse serum. So for this he got the Nobel Prize. What are the advantages and the disadvantages of the method? It is very useful in the biochemical investigation. The biological molecules can be easily analyzed with the accuracy by the electrophoresis. The small quantity of the sample can be analyzed by the method. So this is again an important advantage of the method. And the method is of low cost and easy maintenance is there. Now if we talk about its limitations or the disadvantage, it is unsuitable for the accurate mobility and the isoelectric point determination. So the isoelectric point of the amino acids separation, that creates the problem. Due to the presence of the supporting medium, the technical complications such as capillary flow, electroosmosis, adsorption, and molecular sieving just affect the results. So these are the limitations of the method, but it is very easy to separate out the ionic biological molecules with the low cost, and it is very easy to maintain it. Under the influence of the electric field, the ionic molecules or the charged particle migrate towards the opposite charge of the electrode. So the rate of the migration of an ion in an electric field depends on the number of the factors. What are the factors which influence that rate of migration? It is the net charge of the molecule. So if the net charge is higher, the higher will be the rate of migration. So it depends on the net charge of the molecule. It also depends on the size and the shape of the particle. If the size is smaller, the particle speed is higher. So smaller the size, the speed is higher and higher rate of the migration. The different shape also affect the rate of migration. If the shape is, we have a rod shape or the spherical shape, it also affects the rate of migration. The strength of the electric field, it is also the factor on which the rate of migration depends. If higher will be the strength of the electric field, higher will be the rate of migration. The properties of the supporting medium also influence the rate of migration of an ion, because supporting medium can increase the rate of migration as well as decrease the rate of migration. So when the supporting medium allows your ion to move with a greater speed, it involves the higher rate of migration. So there the supporting medium favors. If the resistance is higher by the supporting medium, or we can say the adsorption on the medium occurs, then the rate of migration reduces. So with the property of the supporting medium also, it is very much affected the migration of an ion. The temperature of the operation is the another factor on which the migration depends. So at a normal temperature, the velocity of the movement or the speed of the movement is normal. If the temperature increased, the rate of migration increased. If the temperature decreased, the rate of migration decreases. What is the principle involved in the electrophoresis? Any charged ion or a molecule migrates when placed in the electric field, and the rate of the migration depends on the net charge, size, shape, and the applied electric current in the form of electric field. So it is totally based on the Coulomb's law and the frictional force generated by the molecule when the external force is given. So here comes, if we calculate the Coulomb force and the frictional force, so it comes out the V is equals to EQ upon F, where F is the frictional coefficient. This F is the frictional coefficient which depend upon the mass and the shape of the molecule. And E is the electric field, Q is the net charge on the molecule, and V is the velocity of the molecule. So as the rate of migration depend on all these things like charge, size, and the shape and the applied electric current, so this comes V is equals to EQ upon F. What is the role of the buffer solution in the electrophoresis? The electrophoresis is performed in the buffer solutions to reduce the pH changes due to the electric field. When we apply the electric field, the pH should not change. That's why we use buffer solution, which is important because the charge on the DNA and the RNA depends on the pH. But running for the too long can exhaust the buffering capacity of the solution. So the buffer solutions are used to resist the pH of the solution under the influence of the electric field. So because the charge on the molecules or the ionizable groups depends on the pH. Now we see the different type of the electrophoresis method. We can classify the electrophoresis into two main types. The first is the free electrophoresis and the second one is zone electrophoresis. The free electrophoresis, also called frontal electrophoresis. This type of the electrophoresis occurs in the free electrolyte solution, while the zone electrophoresis occurs in the supporting medium. The supporting medium, under the influence of the electric field, the method is used zone electrophoresis, and the free electrophoresis, it occurs in the free electrolyte solution. Now we see the free electrophoresis can also be divided into two types. The one is micro electrophoresis and the second one is moving boundary electrophoresis. And the zone electrophoresis can also be divided into three types according to the supporting medium used, that is paper electrophoresis, where the paper is the supporting medium. The cellulose acetate electrophoresis and the gel electrophoresis, where we have the gel polyacrylamide gel, which is used as a supporting medium. So on the basis of the type of the electrophoresis used, according to the electrolytic solution or the medium, we can understand different type of the electrophoresis method. So we have to take one by one the free electrophoresis and the zone electrophoresis, including the main principle, the instrumentation method, and their advantages and the disadvantages. So according to the supporting medium, we have classified the electrophoresis into the different phenomena. In the zone electrophoresis, we have paper, we have gel, we have cellulose acetate. So the role of supporting medium, how it plays. A supporting medium is a physical support through which the charged molecules get separated. So it allows the charged molecule to get adsorbed over this surface of the medium, which helps to separate the molecules. It has two primary functions, that is adsorption and the molecular sieving of the taken molecules which are intended to be separated. So it easily goes on adsorption, where it goes for passing to the medium and separated out. So let's see the moving boundary electrophoresis. It is the type of the free electrophoresis, or it can also be said as a frontal electrophoresis. What is the principle? The moving boundary method allows the charged species to migrate in a free moving solution, that is electrolytic solution, without the supporting medium. So here you can see this is the moving boundary, which consists of the U-shaped glass cell. So always moving boundary electrophoresis is like this U-shaped glass cell of rectangular cross section with the electrode plates, one each of the limb of the cell. So we go for connecting with this U-shaped glass cell the two electrodes, and the sample solution is introduced at the bottom or through the side arm. The apparatus is placed at a constant temperature at 40 degrees centigrade. So it goes for allowing the migration of the charged particles through this glass cell, and with a constant temperature. The detection is done by measuring the refractive index of the solution, and that can be through the Schlieren optical system, which goes on detecting out the various molecule present in the sample, the refractive index of whole solution, and can be seen according to that the molecules should be separate out. So here the U shape you are seeing, there is a resistance towards the moving boundary which causes the migration of the particles. What are the advantages of the moving boundary electrophoresis? Biologically active fractions can be easily recovered without the use of the denaturing agents. So the active fractions can easily be separated out. A reference method for measuring the electrophoretic mobility. The minute concentration of the sample can easily be detected by the moving boundary method. For example, 0.05 milligram per ml, if the concentration of the particle it is there in the sample, it can be easily detected out by the interferometric optical system. What are the disadvantage of the method? It is costlier method, the whole equipment or the instrument, and the elaborate optical system are required to detect out the sample separation. The application, if we go for seeing to study the homogeneity of a macromolecular system and analysis of the complex biological mixtures, this technique is used. The capillary electrophoresis is the type of the moving boundary electrophoresis where we have taken a glass tube U shape. There you can have the capillary of the typically 50 micrometer with a diameter 0.5 to 1 meter in length. So it is the type of the frontal electrophoresis, the pre-electrophoresis moving boundary we have talked about. So just see how the capillary plays an important role in the method of the separation of the charged particles. The principle behind the capillary electrophoresis is that the charged molecules will migrate towards the opposite pole and separate from each other based on their physical characteristics, like its size, shape, and how much strength of the electric field is applied. The capillary electrophoresis has grown to become a collection of a range of the separation technique which involve the application of the high voltages across the buffer filled the capillaries to achieve the separation. So here it is specified that the capillary is used where the separation takes place, which involve the high voltage application, and the buffer solution are required to resist the pH. Capillary electrophoresis is the technique of performing the electrophoresis in buffer filled narrow bore capillaries, normally 25 to 100 millimeter in internal diameter. In this way, a high voltage is required, that is 10 to 30 kilovolt. So under this high voltage, the separation is carried out. The capillaries are typically 50 micrometer inner diameter and 0.5 to 1 meter in length. Now, what's the main thing require here for the separation? Due to the electro-osmotic flow, all the sample components migrate towards the negative electrode. The capillary can also be filled with a gel which eliminates the electro-osmotic flow. The separation is accomplished as in a conventional gel electrophoresis, but the capillary allows the higher resolution, greater sensitivity and online detection. So we can use a supporting medium gel also in the capillary electrophoresis, but without the gel, the capillary allows the higher resolution, the greater sensitivity and online detection. The capillary is filled with the electrolyte solution which conducts the current through the inside of the capillary. The ends of the capillary are dipped into reservoirs filled with the electrolyte. Electrodes such as platinum are inserted into the electrolyte reservoir to complete the electric circuit. So, it is the high resolution separation technique conducted in a solution having the buffer in a narrow capillary tube of the specific size like diameter and the length, and high voltage is required 10 to 30 kilovolt. On the basis of that, the separation takes place. So this is the capillary according to the specified size with the diameter and the length. This is the capillary, and we have the two electrodes, cathode and the anode here. So the buffer solution is there to maintain, to resist out the change. So here is the light source, the photocathode, then data is recorded in the computer monitor. So we have this type of the graph. The sample application is done by the high voltage injection. The potential is applied causing the sample to enter the capillary by the combination of ionic attraction and electroosmotic flow. The second type of the application is pressure injection. Pressure difference is used to drive the sample into the capillary by applying the vacuum. When potential difference is applied, net migration occurs in the direction of cathode. So all the movement of the ions move towards the cathode due to the phenomena of the electroosmotic flow. The neutral molecule move at the same speed as EOF, that is electroosmotic flow. The positively charged species move faster. The speed is sum of the EOF and electrophoretic mobility. Negatively charged molecules lag behind. What is electroosmotic flow in the capillary electrophoresis? The surface of the silicate glass capillary contain the negatively charged groups. So capillary always having the negatively charged groups that attract the positively charged ions. The positively charged ion migrate towards the negative electrode and carry the solvent molecules in the same direction. This overall solvent movement is called electroosmotic flow. So here you can see in the capillary, this is the cross section of the capillary. Capillary is having the negatively charged functional group. So the positively charged groups migrate towards the negatively charged group, and overall the movement of this solvent towards is called the electroosmotic flow. During a separation, uncharged molecules move at the same velocity as the EOF with a very little separation. The positively charged ion move faster and negatively charged ion moves slower. So on the basis of this, the negative ions and the positive ions can easily be separated out. So the electroosmotic flow is having in the capillary electrophoresis, which allows the separation of the different charged particles in the capillary. A small volume of a sample is moved into one end of the capillary. The capillary passes through the detector. So the detector is there, which usually having the UV absorbance detector at the opposite end of the capillary. The application of the voltage causes the movement of the sample ions towards their appropriate electrode, usually passing through the detector. A plot of the detector response with the time is generated, which is called electrophoregram. So that is recorded in the recorder, by which we have the electrophoregram according to the detector detected according to the time. So the faster particle, when it reaches it having the less time, and then the slower particles move towards it, it goes on recording with the longer time. So by that we can determine the molecules. So this is all about the capillary electrophoresis. In the next lecture, we should cover the zone electrophoresis method. Till then, thank you and take care.